Linda's in the chat. Loud place is loud. I think folks will keep joining us. I'm going to go ahead and share my screen on the agenda. Welcome, everybody, to our monthly meeting. We'll see folks who pop in here as we go. Let's see, we got Giovanni here. And Michael is showing up late. He'll be here shortly. We've also got Bradley from the OpenWorm project joining us. I feel like it's good that we continue to incorporate him and those efforts. So, join me in welcoming him. Hey, Matteo. Hey, Porig. Hi, guys. You guys have just joined. I will. Oh, everybody's in a loud place today. I will paste the agenda again. Okay. So the updates I want to give. There's actually a lot going on, and I'm, as tempted as I am to like steal Travis's thunder, I will avoid doing that, and I will just wait till he updates, because there's a lot of stuff going on in that whole camp. But I will just hold my tongue, and I will just update on things that I can uniquely update on. So since the last meeting, we had attendance at the International C. elegans meeting. Actually, it was myself and Balash who was there. So that was actually a very good experience. I just wanted to update on some of the things that came out of those interactions. Hello, Andre.
Hi, guys.
Just updating real quick on the International C. elegans meeting that I attended. So a few things that came out of that. So it was at UCLA in Los Angeles, and one very interesting interaction came from meeting a group that's a group of undergrads that has been formed by a charismatic and enthusiastic physics professor named Katsushi Arisaka. He originally did work in particle physics. In fact, I think his work actually led to discovering of some basic particles a while back. But he, since 2013, basically since the Higgs boson was discovered, he decided he was done with particle physics for the moment, and he decided to form this group of undergraduates, of which there's about 30 or 35 folks, and their mission is, as an undergraduate club, to study the C. elegans. And so he has actually gathered about 4 or 5 rooms in a study hall in UCLA campus, and has set up a whole bunch of microscopes ranging from simple to very sophisticated, and has rallied this group of undergrads to do basic research on the worm. Now, one of the interesting things about him, as opposed to other C. elegans researchers, is that he's coming at this from a physics perspective, and he actually likes reproducing other people's results and getting the undergrads that he's working with to reproduce results. So he's kind of rediscovering a lot of the experimental results, within the C. elegans research world. So I presented OpenWorm at this group. I handed out a bunch of stickers. They went very fast, and Balash was there with me as well. And so we both sort of talked about OpenWorm. It turns out that one of the images that has come out of the project is actually the iconic image for the group. So our NeuroConstruct connectome actually shows up on the upper left-hand corner of all of their posters. I don't know that there's a really good image of it here necessarily. There might be maybe further down. There's a poster image, but anyway. So it turns out that there's a lot of interesting energy and possibilities between these two groups. So we've been talking to them on multiple fronts. We've opened up a line of communication to have some of them play around with C302, and one of their students, James, is trying to load that up and try to basically play around with the neural network. There's another line of interest on being able to get set up to potentially generate any new movies that we would want to feed for experiments. So if we want to do like a touch experiment or something, it's possible that some of the undergrads in this group could collect data and collect a movie that we ask for and send it over to us as part of their undergraduate project. And then a third area of connection is that we told them about the Worm Sim, and we know that one of our missions with the Worm Sim is to make it available to a classroom. So it would seem like a very good idea to share this with them and have them give us feedback and play with it and help improve it, because they are, like I said, studying C. elegans on a regular basis. And a fourth area of interaction that's been interesting is that the folks interested in DevoWorm, so doing the development of the worm, are in conversation with these guys to collect new movies that are created to specification of the dividing embryo over time, and they're talking about that. So there's this really cool interaction where students who are studying C. elegans not for class credit, not because they want to publish a paper necessarily, but because they're actually finding it interesting. There's actually some really cool overlaps with what our mission is on the OpenWorm side, to build code and algorithms and pull data together. So they actually presented, I think, 5 or 6 posters at the meeting based on work that they've done. And the leader, Dr. Arisaka, who's he's here in the center of this collage. He's committed to keeping this group going for as long as he's at UCLA. I think he's sort of a force of nature, basically holding it together with making everybody excited and happy to work on worm experiments. So I think it's a really interesting connection. I'm sure if there's any ideas or thoughts that you have about ways to further collaborate with these guys, he'd be happy to hear about it. I think the one thing is, I guess, probably not to overload, because we've got now many, like almost four different possible directions. But anyway, yeah, it's a very cool interaction and relationship, and it's sort of largely moving forward through integrating with other activities we're already doing. But yeah, so I wanted to make that clear to folks. They're doing things like trying to discover the central pattern generator. So I sent a mail yesterday, kind of pointing them to stuff that we've already thought about with regards to that. Anyway, that's very nice. Okay, another thing that came out of the meeting, which is of relevance to us. I unfortunately don't have as many images to point to, but I just want to point out there, in terms of scientific data, what was reported at this meeting in terms of new experiments, the cutting edge of experiments that are coming out right now are really relevant to us. So I'm calling it whole brain imaging and cell ID. And let me just take this off here to kind of illustrate this. So basically, so a year ago, or less than a year ago, we saw articles coming out about recording from all 302 neurons visually using calcium imaging. And at the time, it was sort of comparable to some of the calcium imaging that we've seen in mouse or zebrafish. And I thought that it would be a while before they're able to start really segmenting out into the activity of individual neurons, which is really, I think, the key thing. So we had this journal club last year. We invited some of the folks that did some of the original methods for that, so you'll remember that that's happened. Well, the progress in less than a year on that whole line of research is incredible. And the cell ID part of this is that the methods are getting so good now that they're able to much more easily segment out the activity of individual neurons. It's not perfect yet, and it's not published yet, but it has been presented here at this meeting. And they're able to start sorting through the signals of specific neurons, aggregating them together, and forming what they're calling like a neural manifold. So they're able to basically boil down the activity of 302 neurons into a three-dimensional space using some math known as principal components analysis. So the point is that they're able not just to have pretty pictures, but they're able to actually show the activity of the whole nervous system in a mathematical way. They're able to extract the signals in such a way that they can describe it mathematically and geometrically. Now that doesn't describe it causally, so we still need to be able to connect it with the connectome and the anatomy and all this. But the point is that what's happening here in this field is basically going beyond what's happening in the fly brain, what's happening in the mouse brain, certainly what's happening in the human brain in terms of the level of ability to analyze the signals that are coming out of the worm. And yes, the calcium imaging, the temporal resolution maybe isn't as good, but the point is that this kind of data is the stuff that I think we were dreaming about when we started the project back in 2011 of being available. And now, only 4 short years later, it's literally coming out. And I think if you give this another 5 years, it's going to be incredibly big news in the broader topic of neuroscience, that we are able to do this at the level of a whole organism. So now, happily, a lot of the people involved in this are already friends of the project. So Ev Yemeni, who was responsible for the database of movement behaviors, is leading the charge on cell ID. So he's able to make a worm that has a different color for each neuron, which helps you to distinguish which neuron you're looking at. And if you combine that with the whole body calcium imaging, now you have the ability to do much easier segmentation. Saul Cato, who we've been talking with in the project for quite a while, who's over at the lab of Manuel, almost 302 colors, yeah, basically, or at least enough colors so that neighboring neurons have different colors. So it's this idea that rainbow is what it was called in the mouse, where like if you've got 10 neurons next to each other, all those 10 should be some different color. But if you've got other neurons that are further away, then they just need to be different from each other locally, but they don't need to be 302 just completely unique colors. But yeah, that's the point, is to distinguish. So yeah, it's very cool. And so there was a workshop talking about basically how to combine these methods. So they're not fully integrated, but they are still able to extract quite a lot. I think the top number, I think they're able to extract signals from 150 unique neurons right now. And they'll get to 302 very quickly, and using methods like this, they'll be able to. So bottom line, friends of the project are doing this. So it's being driven by the postdocs that are coming out. And it's a really interesting time for talking with these folks. You know, I have not yet had time to email Saul specifically, get him in for a journal club, because I think it's really of great significance to the community, and then hopefully get him to start sharing his data, because this is exactly the kind of data that I think we've been waiting for. And like I said, I mean, you know, it's the place where the limitation of physiology, where we haven't been able to stick an electrode in the neuron, is starting to give way to imaging, where we can get, you know, at least low temporal resolution signals from all neurons simultaneously. And if you imagine then combining that with what's been going on in, you know, in movement validation and worm tracking, and what, you know, labs have been doing, where they can both see the posture of the worm as well as see the activity of the neurons. Now, again, imagine the significance of this in a broader sense scientifically, where you're able to see a whole organism's nervous system as you're watching its behavior, and with the statistics that you can pull out of its behavior as well as the neurons. This is what I'm talking about, that like in 5 years, I think these postdocs are going to be standing up at the Society for Neuroscience meeting, giving keynotes to the entire neuroscience community about how the insights that they've been able to get, because you just can't do that for any other organism. So anyway, the significance to OpenWorm is to move our methods forward so that we can support these data collection efforts, and so that we can constrain our models using these data, so that we can really, you know, do what we've been wanting to do, which is to help explain locomotion, to help build models that are, you know, working in partnership with experimentalists and this sort of thing. So there's a lot of reasons to be really excited about what's going on in the C. elegans field, and you know, I don't tend to be taken to exaggeration or getting breathlessly overexcited about data, but in this case, I think it deserves it. So so yeah, any questions about that, guys? Anybody want to say anything about that?
That sounds really good. I'm assuming that any of this data that was not published yet isn't available online or any slides or anything like that. But if you could maybe suggest, I mean, talking about this analysis with neural manifolds and so on, maybe try to come up with some kind of image or some kind of a sketch that how they're presenting their data. If we can make up some scripts that we can pull out calcium activity from maybe the network model and put it into something that looks similar, then I think that might be a good way to make sure that they get interested in the type of data that we're generating from our networks. And yeah, I mean, make it much easier to compare in the long run once they actually start producing this data and releasing it, what the network is doing and what they're doing. If you can, at some point, have some idea of how we might present our calcium activity data from the neural networks.
That's a very good suggestion, and it inspires me to show you one picture that will give you a sense of what that is. I'm just looking it up on my end. Anybody else have any comments? Okay. Let me just see if I can show this real fast. One key picture. Yeah, okay. Okay, well, this is going to be as close as it gets over here. So work the way that they've been presenting data is similar to work that's already been done in both for a much smaller set of neurons in the leech and the locust, work by Brigman and Christon, and work by Gilles Laurent. And this is the closest I can get right now. I'm going to show you something that looks more like data. But basically, it's being presented very much like this, where there are trajectories in a three-dimensional space, and the line is indicating the sort of dynamical position, which is sort of the state at one point in time. So this is independent of time. Time is evolving as the point moves around the line, essentially. And he's basically got a diagram where, as the activity of all the neurons move into a region, like let's say they're moving over here where the 3 is, he's showing that that's roughly corresponding to like crawling behavior, and there's another part that's roughly corresponding to sort of behavior where it's sitting in one place but its head is moving. So unfortunately, there was nothing. There was no actual digital version of any of this, and they didn't want you to take pictures of slides like oh, I didn't have it, but it's in this kind of form that he's been showing some of the higher level results of processing the.
Okay, that's great. And that was with principal components, or
yeah.
Okay.
Analysis, yeah. Exactly. Yeah. So I actually I don't know if it's super clear, but I made an issue to try and start to fit, try to see if we could make a diagram like this, just have the methods to do the math, to do PCA on a bunch of neurons. I think it's in the main OpenWorm repo, just so we could start to have a visualization like that. Now I think it's a lot more relevant than it was because actual experimental data is getting presented that way. Okay, I don't want to hog the whole time, and that was kind of a while, but seriously, I think it's a really significant development, and I just wanted to make sure folks heard that. Okay, another major thing that happened, there was a workshop on worm trackers led by Andre Brown, and I got a chance to plug OpenWorm to folks coming more from the experimental side, where their interest is in capturing behavior and doing analysis on the statistical features of worm behavior. So I got to plug OpenWorm, I got to talk about our approach to open science in a room full of about 30 or 40 folks who really Some of them have some programming experience, but I think largely the room was mainly just experimentalists, so it was very nice. A chance to plug the project, and thanks very much to Andre for giving me the chance to. As an output from that, there was a mailing list created, not by me, but by some other attendees, and then the list, it's sort of a private list at the moment, so I would invite folks to it, but again, I wasn't the one creating it. But what is public is we started to compile some of the information about what folks might want from a worm tracker. There's a bit of a balance here because obviously, what we want to do is create community, but we can't necessarily, do like open up a whole new avenue of coding until there's, folks that are working on it. But just to give you a sense of the kinds of things that are getting discussed, folks are essentially the goal of the workshop was to try and find some commonalities between the software that seems to get created again and again, which is related to doing computer vision on worms. And what occurred in this thread that was getting written up is folks started to suggest some formats that could be universal, some metadata that could be universal. Folks are starting to suggest JSON as a format potentially for drawing that stuff together. There were some suggestions for image formats. So essentially, we just created a repo where this document could live, essentially like a wiki page. And at the moment, it's a bit experimental as to what the other folks want to work on and how they want to put stuff under here. I want OpenWorm to be available as a rallying point for collaboration, obviously without creating a huge amount of overhead for anybody. But this was sort of a stab at trying to compile some information on that topic and to try and take try to make some relationships that are coming out of that workshop and bring them into the project. Obviously, folks that are generating this kind of data can also be important allies and friends for us on the simulation side, because if everybody's using similar software, then it makes it easier for data integration down the road, and makes it easier for us to test more advanced versions of the model when they're ready. So that's another output from the meeting. Okay, another thing. I promise this is actually a summary. I had a ton more notes, but this is really boiling it down to its bare minimum. So a big deal poster that was prepared and just made ready just for the meeting. This might not be the best representative, but let me just click over here. So a brand new connectome was released at the meeting from the efforts of Steve Cook, who you'll remember we worked with earlier. His lab is the lab of Scott Emmons, and in collaboration and partnership with David Hall. They were very excited because they presented a poster where they believe they have created the most complete connectome of the C. elegans ever released. And we have the first version of it. And so we've opened this issue to start integrating it. It's not integrated yet at all. But we've gotten permission to work with it, to release it. We do have information on citation, so we'll be adding it to our data sources. But like I said, it's not yet integrated. There's actually still some work to do to fully integrate it. But what I want to emphasize is that the PI that has built this, Scott Emmons and Steve Cook, the whole poster was dedicated to why every other connectome up until now that has been produced for the C. elegans hermaphrodite has been incomplete, and pointed out that essentially even the John White stuff, which was great, they've been through the details of it. They know places where, as they've gone back and recounted, they've found up to 60% or 70% more synapses. The pharynx in particular had a lot more synapses and gap junctions than were originally found. There were parts of the dorsal cord that they knew were sort of undercounted, 30–40%, and they found additional synapses they fleshed out there. There's also arguments of how there were gaps as you go down the body of the worm. There were like chunks of the worm that didn't have as many synapses as they needed to have. So they've spent years essentially generating this new and updated connectome. And it's represented in this CSV file. Like I said, we have the first version. It's even up there before it's been up on Worm Atlas. Obviously, we need to make it very clear where this data is coming from. both for them and for us. But they're really excited just to have it out there. And again, the PI basically feels that every connectome prior to this wasn't really adequate for doing the kind of modeling work that we're doing. He feels that this is now finally a connectome that is complete enough to do real simulating work. So so it's very exciting that we've already at least got a handle on it. The plan is to incorporate it into PyOpenWorm so that it can be called out. So we'll update the connectome version that we've had in there before. But it's very exciting, and it's just another great example of us collaborating with this community and being on really good terms. How many synapses total? No, I don't know, but there's a CSV, so you can count them up. Since there's so many ways to count them, I go back and forth on it. You can look at how many rows and lines. I guess there's 7,380 rows in this spreadsheet. So you might say that there's that many synapses, but also another way to count them is to multiply that number by the sum of the weight column. Yeah. So the CSV, the CSV is it's linked in that issue, but I'm also just throwing it here in the chat. So that data is available. But we need to make sure that the attribution is added and all that good stuff, and it's not obvious from the direct CSV, so it's going into the README that's one level up from that. But yeah. Sorry.
Could I just ask about you mentioned whether it's been published already? It hasn't been published already, but what are the plans to publish it? I mean, are they?
They are saying, yeah. So they are making it available pre-publication. They're very happy with that. They think they sort of unofficially the first release of it is this poster at this meeting. So in the abstract book, it exists. And now they're in the process of writing up a full manuscript on it. But I personally spoke with Scott and with Steve, and they're very happy to make it available to us now, even in advance. So it's very nice of them, and they're very much in an open data mindset. Yeah. Obviously, we want to be able to cite a paper as soon as it's ready, but for now, we can cite that poster. Yeah. Okay. So again, I gotta keep going, but again, it's kind of like a lot of really important and valuable stuff for us. Okay, this one I'll just touch on briefly. 2 years ago, I was interested in what's going on with metabolic networks. Why is that important? You'll remember a few years back, we did a journal club with a gentleman named Jonathan Carr, who built a model of Mycoplasma genitalium, basically a microbe that's even simpler than a bacterium. And one of the data sets that he needed as a prerequisite to building the model that he did, which was just of a single cell, but working on the sort of intracellular activity, was a metabolic network model. So 2 years ago, when I went to the C. elegans meeting, I wanted to see, had anybody built a metabolic network model for any cells in C. elegans? And at the time, essentially, there wasn't any. There was essentially fragments, but nobody had really been putting in the effort to do a similar thing. And I remember I probably had a couple of calls with some folks that were trying to do it, or I was trying to understand the constraints, and I looked up some papers. Basically, there wasn't anything. Well, this year, there has been an attempt to do that. And although the authors would say it's not fully complete, there is at least a start on this. I'll have to look through my notes to give you more of a pointer to this, because essentially it was one poster. And I wasn't familiar with the authors prior to meeting them. But I had a nice chat, and I do have an email address, and so I'm going to reach out some more. So I don't, there's a lot going on, so I don't think I have the bandwidth to really build a whole project under this yet. But I just want to throw out there that that level of cellular detail, a bridge between sort of a car model level model and C. elegans neurons, is now starting to be more possible than it was before, and there are folks that recognize the gap. And are working to fill it in. And so it would be lovely at some point to start filling that piece in. This may also actually be another connection point with the DeBoWorm work eventually, which is to understand what's happening at a metabolic level inside cells as they're developing, in addition to what's happening inside cells when they're adult cells. This could also be relevant to ion channels in terms of putting ion channels into the membrane and taking them back out again. That's something that a metabolic network model could help you understand. So this is probably a placeholder until we get locomotion working. But as this gets more sophisticated, I think it's just kind of like a watch this space kind of a point to keep track of. Okay, and then I think I've kind of mentioned most of this. I caught up with Steve Cook. That was the Connectome stuff. Axel Blau is the gentleman from the C. elegans group. They presented a poster on recent work. They built some very fancy hardware. I don't have really time to recap too much of it, but it's interesting how that's going. I think they're looking to present milestones towards the end of next year. So it's progressing along. Obviously, there's different things happening in different areas of this, and this was sort of trying to unify. It was trying to be a unified presentation to pull many of it together. If you're not familiar with C. elegans, it's the project that is EU funded to build a hardware model in FPGAs of C. elegans in various ways. And so we've been trying to find intersection points and variously working with them and having some interesting conversations with them over the last couple of years since we found them. So it was nice to see Axel there. It was his first C. elegans meeting, and I think he was sort of commenting on how much interesting biological details there were to learn about while he was there. We caught up with Netta Cohen, which was good as well, and updated her on a lot of the things that are going on. She was updating me on some things that were happening as well. I need to kind of digest my notes from her as well, but Balazs is also sitting with me there, so he may be able to comment on that. Andre from the Worm Tracker, Ev from this whole brain imaging cell ID, Bradley, who's here in the meeting, and we got a chance to sit down and sort of talk more about intersection points and coordination points with a broader work on the adult. And I'm hoping that we can continue sort of coordinating here at a high level as we go forward. And so there's now a slot on the milestones board for the work that they're doing. So I'm hoping that we can continue to share methods and coordinate on technology as we go forward. They've really they've been putting their progress up on GitHub issues. They're having weekly meetings now, and so there's a lot of good stuff going on coming out of that. Saul Cato I was telling you about with the neuromanifold, and I also spoke with several folks at Wormbase, Christian Grove and Raymond Lee, who works on the ontology over there. Found out a little bit more about how their web-based API works, and just generally kept up good connections there. Last thing, and I'll just leave it to you to click on the link. I think this happened maybe earlier. I think it maybe happened before the last meeting, but I may not have mentioned it. I made some revisions and updates to the OpenWorm docs in partnership with Travis, but also made some other updates, which I'm sort of calling 0.9. So you can have a look at that. I also put in a pull request to update the website to point to some of the new stuff because our docs were getting pretty out of date. Okay, thanks guys. Sorry to natter on for so long, but I spent I think about 3 days, variously in and out at the Worm meeting, and so hopefully 30 minutes of updates is the best I can do to kind of compress all that stuff so that you get the highlights of it. Any comments before we go on to other updates? You all? Okay, great. So let's go to movement validation and hear from Michael.
Yeah. So just following on what Stephen was talking about in terms of meeting with the Brown Lab and going to the tracking workshop and at the conference. Those are exciting developments, certainly of relevance to movement validation. So as far as our effort goes, we had our regular monthly meeting about a month ago, and we're having another one in 2 days. And perhaps the biggest thing that's been done in the last month in terms of visible deliverables is that this statistics code has been ported completely. So now we can produce, in fact, a PDF document, thanks to some work that Chris Lindsay did, that actually is a rough approximation. It comes close to replicating the look of the Schaefer Labs PDF, because if you recall, originally their code eventually produces this 60-page document that compares experiment worms to control worms and shows which features differ statistically in a statistically significant way, to help you identify what the behavioral phenotype is of mutant worms. And so of course our intention is to have such a PDF for version 1.1 of OpenWorm simulation, and version 1.2, and hopefully to get it closer and closer to be matching. So now we have this PDF. So theoretically, you could feed in an OpenWorm simulation list of skeleton coordinates and get out a PDF like this that summarizes the differences. So I think that's a major accomplishment on our end to be able to show that. In terms of other efforts, because there's still more to be done at various parts in the pipeline here, Jim's been working on a refactoring of the features code to be more extensible so that more stuff can be done there. There's more details if you want to talk to him about that. And the computer vision work is being, of course, done mostly by the Brown Lab themselves. We also have volunteer David Clark, who came through the project through OpenWorm, actually, and now he's, as part of his CS Masters, working on a sub-problem of the computer vision algorithm, which is identifying the head and the tail in each frame. So right now, the computer vision algorithm works. It is complete, the one that's developed by Casey at the Brown Lab, but there's more improvements that could be done, and one of them is this head-tail identification. So we're very happy to have the capacity to be able to work on that and to contribute to the effort that Andre Brown's team is working on there. What else? We also have Aidan Rock, who's working with Balash, of course, up there at Edinburgh University, on motif analysis for Balash's work, but he's also been working in the last few weeks on porting Andre Brown's motif analysis code from back when Andre wrote this paper. I think it might have been his PhD work, I believe. So that is in MATLAB as well, and if we converted that, then we'd have an additional capability to analyze the behavioral differences. So that's good, and apparently there's been some good progress there. So you can see from some crossed out items there, we've accomplished several of our next steps, but we basically at this point definitely need to be focusing on getting the package running in the cloud so that we can achieve some of those goals that we've been talking about over the past few months around getting these features and these videos processed in sort of a batch form, so you'll be able to run this package that we've been working on for so long on a huge numbers of videos at the same time, and then producing the result. So one of the potential outcomes of this, for example, could be rerunning all of the Schaefer corpus of videos, and then potentially writing a paper or a letter to Nature Methods giving an update on how, with the improved computer vision algorithm and the improved feature code, how has this changed the statistical results that were previously published 3 or 4 years ago when they originally ran the results with their original MATLAB code. So that's certainly an exciting thing that Andre Brown in fact suggested, and perhaps we can be a big contributor on for that. So that's basically what's going on in movement validation.
All right, awesome. And yeah, did you want to say anything else about these other folks who are coming out of the worm tracker meeting, and just, I don't know, your view on it? I know that it's still not very settled or clear, maybe what direction folks are taking, but you've seen a little bit of activity on that.
Yeah, I mean, I think it's exciting, even if their involvement is only just offering the suggestions that they have so far, and maybe a few more. Even if it's just a matter of the offering suggestions, it's great because it gives us feedback to be able to make the open source code be as usable as possible, be as forkable as possible, if you will. So someone is going to want to potentially use it. If we're building this thing only with the OpenWorm use case and maybe also the Brown Lab use case in mind, then it's going to be probably less useful. So it's really great to have the feedback of the broader community. And it's also heartening to know that some of these same issues that we've been grappling with in terms of figuring out what the specification is, the metadata requirements, whether to use chain code lengths for calculating the skeleton, things like this. It's heartening to know that these are problems that are faced by all these groups for sure.
Cool. Right on. Okay. Shall we go next to Geppetto?
Hello, can you hear me, first of all?
Yep, but you got a good deal of background noise. You probably want to just like with your mouth right in the microphone.
Okay, I'll try to do that. So yeah, bandwidth is not too good. Let me know if at any point you cannot hear me. So yes, update for Geppetto. There was since this meeting the release of 0.2.4. So I'm going to, it's already linked on the agenda, but for anybody that wants to take a look, I'm also putting it in the chat. So 0.4 was a rather important release because so many features went in. And it also took more than it usually does, which is a month. I think this one went above 2 months, but integrating all of this was really a lot of work. So if we look at my screen, so that if you guys can see it, I will go through some of the features. But so I won't name all the contributors, but you can see their names in this list. So thank compression of basically anything that is sent from the server to the client, which helps improving. Do have a dashboard that I will show in a second that allows to interact with what we are now calling Geppetto projects, which is something more than what we had before, which was just a simulation. But I won't go into many details here. I will say something during a brief demo. Next, we have now enabled or running neural simulations using Neuron, which is something that we wanted to do for a long time, and now finally it is added, and we refactored all the user connection. There is now the ability to change the simulation parameters and basically run the simulations with the new parameters and see what happens. There is support for a database. There is support for a database and for S3 that these are optional modules as it's written down in the Oh, okay. Nobody can see my screen for some reason.
Well, apparently Corey can.
Oh, sorry, I was looking at yours.
Okay.
Oh, okay.
Oh, okay. Can you see my screen? Or shall I try to refresh? Or
I just see it.
maybe shall I? Oh.
Maybe you should refresh.
I'll try to refresh. Otherwise, I'll abort the experiment.
Yeah.
Okay.
Okay. Okay.
It's a big improvement. Now we can't see him or hear him.
Oops. Okay.
I think that's it.
Buffering up.
There you go.
Ah. Well,
I'm getting a weird error from Google. I'm getting this error. There was an
I can see
error
your screen
while
now.
loading your app. Can you see my screen?
Yes. Yeah. Try to continue.
So can people see my screen? I know Stephen, you can, but can other people see my screen? Maybe.
Yes. Yep. Yep.
Okay, so yes, I was basically running through the feature here in the release notes. We added support for a database which basically allows, by deploying an additional Geppetto module, to have users basically saving their workspace, what projects they are working with, what models they are working with, what simulations they're running, etc., etc. And there is now the ability to download models. So you're looking at something and you want to download it, so they should
Hello.
download the model that you're working with, as well as the ability of downloading the simulation results. So it's not only something that you'll be able to look at the web browser, which is good.
You're breaking up, Matteo.
I think the bandwidth is crushingly slow, unfortunately. Up, and there he goes. Okay. Well, hopefully he'll get into a better state here in a minute. I know. I think Giovanni might be in the same place, but all right. Let's. Yeah. Okay. So I think he's going to keep working on it. Let's keep going on some of the other topics then. So let's see here. I don't think so. OpenWorm update I think can come in with the rest of the sort of cell modeling stuff. So let me. Oh, okay. Is that Travis? Did you put these two down? Is that two things you put down?
I did not put those there, no.
Okay. Okay. Well, let's have your update anyway. And,
Yeah, sure. I mean, that is basically my update, so thank you to whoever put that there. Okay.
Is it?
So, sorry. What was that?
That's your update, but you didn't put it there?
I mean, it's basically what I was going to talk about, like very similar.
Okay.
Yeah.
All right. Cool. It's probably from last time. Then.
Oh, is this an old agenda or something? It's
Well,
gotta
what
be
we do is we copy the old one into the new one.
Oh,
So,
well, okay, well, I was wondering which benevolent person did this, but yeah, I guess it was me a while ago, and I just don't remember. No, yeah, okay, it's got to be mine. And whose else would it be?
fair enough.
Yeah, okay. Well, anyway, yeah. So, I guess my update is basically since the last OpenWorm General Meeting, we've had 2 hackathons, and they've both been successful. I guess in terms of people came and enjoyed it, and we did get some deliverables, like some code at the end, and there were issues resolved. So in terms of attendance, there were 10 people at the first hackathon and 11 at the one a few days ago in total. So I mean that was like really encouraging to see that people were willing to donate their time on a weekend to pop in and just even just to have a chat in the Gitter chats, or to actually contribute some code and start pushing things in the modeling section forward. So we have set up like basically 2-week intervals for these hack sessions. So we'll be having another one. We had our last one on this past Sunday, so we'll be having another one in a couple weeks. So it might not be on a Sunday. That's not a good time for everybody. But we'll send out a poll for the next one as well and go with most popular time again. So yeah, I mean, big thanks to like everybody that came out. That was awesome. In terms of progress on specific sub-projects, at the hackathon there was some work as well with the ChannelWorm web interface. So I'm just going to share my screen and show you what that looks like right now. So it's a curation interface. Just log on to this URL. You can get it from the ChannelWorm GitHub repo. And then in each of these side panel, in this nav bar, basically you can go to different data objects and enter data from a paper. So the idea is basically that you can go from a paper to an ion channel model entirely now within this single unified interface. So over the weekend, Srijoy, Vahid, and Milad, among others, were present at the hackathon, and there are actually some data entered in this now. So we actually do have some plots from the literature that are digitized. We have data in this database, and we're using it as a curation interface. And I guess one important thing to know here is that the users. So for instance, Srijoy noticed some things on the weekend that were kind of clunky or difficult to use from a curation point of view. So he informed Vahid and Milad. Then basically the problem was solved instantly. So basically the point I'm trying to make is that we've got this rapid feedback development loop going on right now, which is basically evolving the user interface for the use case. So it's pretty exciting to see that kind of thing. And I mean, the UI looks great and all that stuff. So big thanks to Vahid and Milad and everyone else that's contributed to that. What else do I have here? Okay, well, I was going to talk about the connectome data, but Stephen mentioned that. So that's also really exciting. That's going to be a big development in PyOpenWorm. Right. Okay, and also the unified model completion dashboard that was mentioned in the last OpenWorm general meeting. Over the weekend, Joe Bowen started a new repo. He has a new cloud project on OpenShift, and this app is up now in terms of you can access it via URL. There's an actual space for it. So now we just have to populate it with contents. So that's going to be, I guess, most generally a web interface and a high-level interface to interact with and see the levels of completion of different models that we're going to be storing in or around PyOpenWorm. There is some talk now, of course, about the separation of modeling data from like actual known data in PyOpenWorm, but that's a separate issue. But this interface is going to be for wherever we store the models, whether it's in PyOpenWorm or completely separate from it. This will be like a high-level interface to it, a web interface to it. So that got started over the weekend in the hackathon as well. So I believe that's it for my updates.
Great. I was chuckling to myself because I said I wasn't going to steal your thunder, and then I still did.
No, that's okay. That's okay. Okay.
But yeah, no, it's great work. And in addition to those folks showing up at the hackathon, Travis is regularly working with folks that are there. You know, the testing suite for PyOpenWorm has made leaps and bounds based on his work with a gentleman named Gopal Sarma. And that's starting to snowball out to some other folks that are contributing, like Chi Lee, who has joined us last couple times in this meeting, and some other new folks that came in. By the way, parenthetically, that hackathon gave rise to the first ever OpenWorm Singapore in-person meetup. On Tuesday, two individuals, we discovered that a new contributor who only came on board last week happened to be backpacking through Asia and was staying at a hostel in Singapore, and Chi Lee, who joined us earlier, is living in Singapore, and so we made the connection only through OpenWorm, and they got together and had coffee and talked about PyOpenWorm testing. So we now can rack up a in-person meetup of OpenWorm in Singapore as a result of our stuff. So I thought that was really cool. All right. So Travis is doing a great job. Really appreciate it. Awesome stuff. Okay. I wanted also here, and so stuff is looks like the document is sort of populating. So sort of I think I'll break from that while folks are making updates. I wanted to give Bradley, Alicia here, a chance to tell us just new stuff in VivoWorm that's been happening, and I'm hoping that obviously again, just trying to get an appreciation for what that sort of work is looking like, and what stuff is going on these days, and so folks get a sense of where it's at.
Yeah, so I gave a journal club last September, and if you want to go back and look at it to see what the project is about, please go back and look. But in a number of different avenues, it's it's starting. We're trying to establish some work so that we can actually produce something. So we're not really at the stage of producing deliverables on a regular basis, but we have a couple of avenues of research that we're doing. One, we're sort of bringing together different data sets. So we've gotten data sets from the worm guides. We've gotten other data sets that involve gene expression and other things. So the idea is that they have a method where you can take an embryo and follow it out to a certain, until it hatches, and you have all the cells from the single cell to, following out through the C. elegans lineage tree, and you can actually find the position of the nuclei in that, in the embryo. So we can actually look at what cells are dividing where, what their position is in the embryo, and we can say things about development. But we also have another opportunity in that vein, which is Dr. Arisaka at UCLA, which Stephen mentioned, and he's of course interested in imaging different biological things. And so I think one of the things that we're going to try, and this is in the next couple of weeks, is he's going to try to image an embryo from the single cell stage up, ideally to the adult, but we're still working out the details on that. He's going to produce, it's like from a single cell to 32 cell stage. He's got this, the state-of-the-art imaging technique that he uses. So you get, you use a GFP marker, and you get very high-resolution images of the entire embryo in 3D, and actually in 4D, since we're going to have a time series. And so this is big data. This little bit of the worm development is going to be like, I don't know, he estimated at 2 gigabytes of data just for that. And so we'll see what we can do with it, but that's an ongoing thing. We also have a purely computational modeling component of the project. So that involves this project that we're calling Morphozoic. So there's a collaborator, Tom Porges, and I think he's worked with the OpenWorm group before. And he's come up with this model. It's a cellular automata. It's where to work on this for. In parallel to that, we also have some connections, and this is something I would like to develop further. The platform CompuCell 3D. So CompuCell 3D is actually an agent-based model. It's not a cellular automata, but it uses the same principle. You have cells that form a grid or form a structure, and those cells represent several weeks ago, and they were showing me how you could model development, you could model physiology, like the physiology of the liver, using this model. It's a physics model, so they use there's a physics involved, but it's more biologically plausible than a solar automata. If anyone's interested in finding out more about that, contact. So I can send me.
Okay, I think we're all experiencing the same thing. Man, this is not our lucky day with bandwidth on the meeting, unfortunately. So Bradley, going to give you about 5 more seconds. See if you come back on. Nope. Okay. Yeah. Not. All right. Well, we got a sliver of it. So we got a sliver of Geppetto, which is that release 2.4 is out, and there's a release notes. Ted's going to make some nice stuff. There's a lot of really great features on that. We got an update from DevoWorm. I also was updating in the agenda. It looks like Mateo's also added to the agenda. And let's see, Bradley, you back?
Yeah.
Okay. Seems like bandwidth is still a little low because I'm hearing you breaking up audio-wise. Try one more time. Okay.
Yeah, I'm back.
Oh, okay. It was a big delay. You want to just wrap up then, real quick?
Oh, okay.
Yeah.
Yeah. So, like I said, So I have the link to the project website up here, and we have the two main foci of the project. The one with embryogenesis, the early modeling side, which we're trying to use them to sort of get insights across methods. But it's like I said, we have collaborators coming in out of the project. If you're interested in contributing or have any ideas, let me know.
Awesome. Okay, very good. Great update. So let's do WormSim update next. It's just coming up on the agenda. Giovanni, see if your bandwidth is any better.
Sounded.
Yes. Okay. Should I try to update? Okay. I'll narrate it. Okay. I see. Okay. So I'm looks like I'm tapped to try and provide the update. So WormSim update. So there've been some latest progress on WormSim, and it's looks like it's manifested itself in a couple of videos. So here's one. So in this video, oh wait. What you're seeing is a loop that's been created between the worm crawling, sort of in this direction, as well as the ability to see 2D graph of one of the muscle cells in a sinusoidal activation pattern. So this is representing the mechanical model. And this is happening in the browser, built in. It's being built on top of the latest release of Geppetto, so it's been coordinated with that. And so that's one latest. And then let's see. Okay. Yes. And yes, as Giovanni's pointing out in the chat, that's coming from Andre and Sergey's cybernetic recording. This has been a process of basically taking the particle simulation and putting a skin around it, and so building a colloidal skeleton out of it and animating that colloidal skeleton. Oops, I don't think that's going to work very well. Animating the colloidal skeleton. So here now this shows you the X-ray view where we made the cuticle transparent. And so inside the cuticle now, you can actually see the muscle cells. It's very nice. And the rows of muscle cells as they were laid out, again, defined by the SPH simulation. They've been mapped to each of the 95 body wall muscle cells. And again, this is operating the browser. You can see in this demo, you can pause the simulation. You can rotate it around so you can see individual muscle cells. So this is very nice, starting to look very much like the worm browser, except it's a worm browser plus it's animated. So crawling worm in the browser, I think it was a goal we came up with maybe a couple years ago that we wanted to see, and it's really quite nice now, this progress here that's coming out. So you want to type anything else in the chat I should mention there? Or does that pretty much capture it? Okay, so almost there. Looks like he's saying should deploy and give access to the OpenWorm team soon. And then around the corner is the release to the backers. So we'll give folks here a chance to kick the tires, check it out. And yes, I'm probably not doing it justice. That's right. So Mateo's pointing out that video is about 3+ years of work when you think about where all the different pieces that we started. And we started with WebGL and Geppetto a bunch of years ago. And it represents also, the building on the foundation wouldn't have been possible without, obviously the building of cybernetic, the creation of the muscles of cybernetic, making that whole animation work, additional novel algorithmic work on how you extract a skeleton from a particle simulation was done by collaborator Martin Madaris. Actually wrote up a PhD thesis on this topic, and we managed to get him to do it for this piece. So again, I know I'm not doing it enough justice here, but I think this is although it looks cool. I think yeah. And thanks for reminding me, guys. It's really important to point out that there's a ton of work that is represented by those images, and really a huge thanks to Giovanni for coordinating it, Matteo for the Geppetto infrastructure, obviously Andre and Sergey for the original videos, Martin for that work and all the effort, obviously of the folks that have been pushing OpenWorm forward that we've worked with up till now to make that possible. So it's really quite exciting, and the proof in the pudding obviously will be the final release, but I think these images show extremely promising progress on that stuff. So,
can I ask a question?
Yes.
So the collada recording there that you're displaying in the browser, it's against a black background, so it's perhaps hard to tell in absolute coordinates where the worm is. So maybe against a grid or something to be able to see. So like the worm as it's moving, you can see that it's actually in fact moving, and it's not just the camera following it. For example, I realize that we're not at the point where it's quite moving around a grid yet, but perhaps that's something to think about. I'm sure it's easier than creating an entire worm to add a grid there, but I'm not going to say it's. I don't know how hard it is, so I'll leave it to you guys. But that'll be necessary, I suppose, to eventually have that connection point with movement validation where we have the coordinates of the worm actually sent over.
Yep, and it looks like these guys are typing here, basically saying you can use whatever colors, you can create videos from that with a grid, to be the input of movement validation, and I think they wish.
Cool.
But,
would it not be, I mean, in terms of actual, the hardcore kind of major analysis for movement validation, would it not be better to use cybernetic directly, the output of cybernetic directly, rather than going through Geppetto in some way? I mean, you don't necessarily want to do everything online if you're going to be running large number of simulations.
Yeah, I agree that, and we've thought about this for sure, that probably doesn't make sense to go all the way to the point of having a video, although that is an option, as Matteo says. It might be a lot easier, obviously, to just get the list of the coordinates frame by frame.
Yeah, and I think to a certain extent that's what they do.
Can you hear me?
Yep.
Yes.
I'll let you know if we break up.
Okay.
Don't break up with me.
So it's not a matter of being online, obviously, for movement validation. It's not about doing this with a in case of movement validation. It's that the video that it matches that.
I'm hearing something that sounds like Mateo, but definitely not hearing any words. It's kind of like the Minions speak version of Mateo.
Yeah. Sorry, you can't unfortunately
Sorry,
understand you. I know, it's very frustrating. Okay, it's not about being online per se, but rendering particles is harder than dealing with a mesh, yeah.
Yeah, that's a fair point.
There's a ton of, I think there's a ton of additional design decisions that are going to come out of this FormSim stuff, and I think this is a healthy beginning to those things. The beauty of making progress is that immediately once you see the progress, we're now able to think creatively about next steps. I think one thing that I was hoping that Mateo could get to, but I didn't get a chance to mention, was that we were looking at that integration diagram that he put together, and some of you may have seen it come across in GitHub issues, with a big box diagram that's pulling all the pieces together. And so right now there are some like parallel lines that represent potential options for flows, so that one path may be a direct from cybernetic, another path may still go through the animation meshes. I think the point here of this open source community is like not to mandate one way or the other, but to provide options, and then hopefully folks will sort of try to work on whichever one they think is the most promising for that path. So I think both. I think there's a good case to be made for both, and I think it's exciting to think about how we might move that forward. So I think Mateo is starting to think about a path that maybe does still involve the skeletons, but I think there's pros and cons to either. Oops, looks like we're losing everybody now, and Andre was going to be one I was going to get an update from, but okay. So anyways, chat's going forward. Let's hear from Porig because he seems to have good bandwidth.
Yes, but unfortunately I don't have too many updates. I have been doing various random things on different projects, including DNRML and some things related to the IV curve generation. Hopefully most people have seen some of those through the issues and so on. I've done a little bit with C302, but nothing worth any release just yet. And a little bit with cybernetic, but again, nothing to point people at. I'm hoping computational neuroscience meeting is next week. I'm going to be away for that, but hopefully after that I'll have a little bit of time to maybe wrap up some of these threads and hopefully make another more stable release of C302 that integrates some of these latest developments, and also this network tuning script is coming along nicely, and you can tune the behavior of the network, but it's not tuning well at the moment. It's still, but the overall framework is there. So a few different things, but nothing that I can point people at and say this works.
Okay. Yep. I do just want to highlight that a little bit more, because I think you're underselling a little bit of the effort, which has been really excellent. So Porig has organized a workshop at the Computational Neuroscience meeting in Prague next week, where they'll be presenting a bunch of different things. They'll be talking about recent progress of the brain, which is obviously relevant to the group. They'll be talking about some of the updates to Geppetto that Matteo was talking about, and then Matteo will be presenting 15-20 minutes on recent progress in OpenWorm. So you guys will both be there. And I would just, and then Porig also took the lead on the poster. There's a poster presentation going in there. Thank you very, very much for doing that on behalf of the project. And it's looking quite nice. So I would just ask you guys, since there's the opportunity really to interface with the rest of the community there at CNS, that if you guys can just take down notes of any relevant interactions that we might bring back into the group. It'll probably be a whole month, right? So until this next check-in, so we may not get a chance to talk about it unless it's something that's really exciting. So, but just like jot down some notes, both of you guys, so that you can recap the way that I was starting to recap on the exciting bits for the Elegans meeting, and I think that'll be to everybody's pleasure. So yeah, so that'll be an awesome meeting. It's always been a good meeting in the past.
Yeah, no, it's going to be a pretty good meeting, and hopefully that workshop is generally on open collaboration, computational neuroscience. Some of the Human Brain Project people, Sri Jordan there, and Alan Brain Institute, so, and also the neurokernel Friday people. So hopefully there'll be good discussions there around posters and so on. So yeah, I'll try to summarize some of the outcome of the meeting. Yeah.
Cool. Okay. Well, last but certainly not least, Andre, who we've been busily writing the next version of that paper recently. Please give us other updates on stuff.
Okay, yes. The first part of the work was writing a paper. I hope we'll finish it soon. During this, a lot of useful things were done. One of them, for example, is benchmarking of cybernetic at various devices, CPU, GPU. How performance depends on a number of particles. For example, we found that we can simulate 1.5 million of particles, both on CPU and GPU. It seems to be an upper limit, but still works at nearly 0.5 FPS, and it's one of the most fastest simulations between everything which exists in the world. Well, the next part is I continue my work on simulation of a single C-against neuron, but very detailed, biophysically reliable, and so on. So, reading papers, extracting important data related to this purpose, and reading the neuron book and techniques, how to work with it, how to implement ion channels and other mechanisms involved into neuron functioning. And third thing, I have an idea, maybe it will be useful. We have C-against body, which can crawl in cybernetic and in Geppetto. Currently, it can perform sinusoidal movement, which causes its locomotion forward. I plan to study reversal of the movement, so trying to make the worm move forward for a while, then change type of oscillation and crawl backward, and also try to check is this worm body able to perform, for example, the turn, not reverse, but turn through making a loop and then crawling backward. Doing it. So try most well-known types of movement and check if this current worm body is able to perform them in our existing model. So this can become a basis for a second paper, which will describe not cybernetic but development of against worm body inside it. But I think it can be also interesting for the overall project. So since the worm body is ready, I just need to develop a number of input for motor neurons, so some kind of how signal at every neuron should evolve with time. I don't think it will take much time, but a lot of calculations. Maybe some videos will be recorded. So that's the plan for the near future. That's it. Thanks.
Awesome. And I wanted to show one thing too, related to the paper. So when we got feedback from the reviewers on the first version of it, they mentioned the work of a researcher named FedQ, I think that's how you pronounce it, at Stanford, who had done some work with coupling liquid and solid simulations. And so this is actually not a movie. This is a simulation of water crashing up against a lighthouse. And for the last several meetings on the paper, we've been trying to understand if this work is, you know, like how this work relates to the PCI SPH approach, because SPH level set is a little bit different. It kind of couples two different algorithms together, whereas we've sort of tried to just use one for everything. There's also some interesting examples. So that's a rigid example. There's some other ones that actually have solid in it. So the good news is that some of these benchmarks that Andre has done has shown that, at least as far as we can tell, we have comparable level of performance to this. And the reviewers were saying, like, well, do you perform as well as this novel method or not? And so now we know that we think we've got a good chance that we do. So there may also be some interesting things to learn from that in the next iteration of the worm body model. But for now, it's sort of driving some good discussion. So yeah, good stuff. And Andre, it's also been good to see you at like jumping into some of the hackathons. So Andre has been joining us on the Gitter chat. If you guys, by the way, have not been on the Gitter chat as much, I think a lot of the stuff that Travis is doing is definitely leading to a lot of conversation on several of the repos. OpenWorm is rather active. ChannelWorm is very active. Muscle Model, so if you want to create a channel for your stuff, it's very easy to do that. If you want to participate in the existing conversations, even if it's on OpenWorm, it's turning out to be sort of a lower barrier of entry than posting stuff on the mailing list. For some certain conversations, plus private messaging is very convenient on the Gitter chat room. I've started to get the DevoWorm folks on that chat room as well. So it's a nice way to kind of drop in on what may be happening just parallel to what you're doing in the project, and maybe a good way to get some sense of integration on that. And obviously that's open to everybody, so do keep in mind that those are sort of public chat rooms, but at the same time, I think you know, a lot of the stuff is very helpful to others. So okay, well, a lot of great stuff in this last month, guys. Does anybody want to make any final comments or post anything in the chat if you're bandwidth limited or anything before we break for this month? Okay. Well, awesome progress, guys. I'm really excited for this next month and the evolution of these next developments. Thank you very, very much for your time here, and I'm just very excited for next time. So we'll catch you in a month.
Okay.
Bye-bye.
See you later, guys.
Bye, guys. Good to see you.